My name is Randy Stout, and I'm an assistant professor at the New York Institute of Technology, College of Osteopathic Medicine. And I'm going to be describing how we're using the grant funding by the American Thyroid Association Research Grant to study how thyroid hormone is processed and transported from cell to cell throughout the body, specifically focusing on the brain. And it's very important to understand how much thyroid is present in each individual different cell type throughout the body, and especially in the brain, where astrocytes are thought to be the main cell that takes up, processes, activates, and then provides thyroid hormone to the other cells throughout the brain. And there are many unknowns, and those unknowns will create a problem when you're trying to judge the effects of thyroid hormone therapy, for example. But we also want to be able to use the tools that we're developing, the research tools, in other disorders such as autism spectrum disorder, epilepsy, and neurodegenerative disorders. These tools will also be extremely useful for the ongoing studies by others at NYT into the effects of thyroid hormone on heart function, heart failure, vascular disorders. And so we modified and improved upon a sensor technology that uses fluorescent proteins to look at the ratio as an indication of the amount of thyroid hormone present in the cells between different compartments of the cell, the cytoplasm and the nucleus. We modified it so that we can also look at the function and morphology of the mitochondria that provide metabolic fuel to the cells. We and others hypothesized that through the control of thyroid hormone, these mitochondrial functions and morphology are underlying some of the detrimental effects throughout the body of hypothyroidism and other changes to thyroid hormone signaling. So we can see here that as we apply thyroid hormone in the top row, when there's no thyroid hormone, the sensor is out in the periphery of the cell. And as we apply increasing amounts, as we go down the rows, it all moves into the nucleus, shown by this blue arrow in the middle of the panel. And that is how we can quantify this effect and then see how much mitochondrial function is in each of these cells. We plan to, using the American Thyroid Association Research Grant Funds, to bring this into animal models so that then we can apply different treatments, including increasing thyroid hormone in the blood supply, and see how that changes thyroid hormone levels throughout the brain, for example, in mouse models of autism spectrum disorder and epilepsy. And so our goal with this tool and the improvement of the tool and the research in general is to be able to understand how thyroid hormone affects the brain and how different treatments for dysfunction to thyroid hormone signaling are leading to changes in the brain and in other tissues throughout the body. Overall, we hope this leads to better understanding and the ability to produce better treatments for hypothyroidism, neurodevelopmental disorders that result from hypothyroidism, but also understand how thyroid hormone could be used as a tool to ameliorate the effects of other disorders such as autism spectrum disorder, epilepsy, and neurodegeneration. So again, thank you for your support. I thank all of the members and donors to the American Thyroid Association, and I'm very excited to continue with this research.